Hello class, this is our recorded lecture about neurotransmitters. We have 4 criteria for neurotransmitters. So a molecule that fits all 4 of these criteria is in fact a neurotransmitter. First, it must be synthesized in the neuron. It must be present in the presynaptic terminal. Its release is increased by calcium in the presynaptic terminal. Third, exogenous administration must mimic the action of the endogenous transmitter. So if we inject some of the molecule into the synaptic space, expose it to the postsynaptic neuron or cell, it must mimic the action of what happens endogenously. And there is a mechanism that exists for removing it from the synaptic cleft. Now let's go over those. We'll start from number 4 and talk about those different mechanisms. There are 3 different mechanisms for removing neurotransmitters from the synaptic space. We have diffusion, enzymatic degradation, and reuptake, which is done by reuptake transporters. First, diffusion. So this is the natural spreading out or away from the synaptic cleft into the wider extracellular space. And we've discussed this in class in the context of concentrations of ions, and we should be familiar with this. This is simply the passive physical random movement of the molecules. It becomes more and more dilute, and it sort of floats away into the extracellular space away from the enzymatic, or away from the synaptic cleft. Eventually, the molecules would be broken down elsewhere. We also have enzymatic degradation, the process by which an enzyme, which is a catalyst of a biochemical reaction, changes the structure of a neurotransmitter so that it is no longer recognized by the receptor. Let's take a look at an example with acetylcholine. So one of the ways that acetylcholine is removed from the synaptic cleft after it's released by the presynaptic neuron is through enzymatic degradation. It's broken down by an enzyme called acetylcholine esterase, which exists in the synaptic cleft. And once acetylcholine is released into the synaptic cleft, acetylcholine esterase breaks down acetylcholine into acetate and choline, 2 different parts. Now that choline is recycled back into the presynaptic neuron through the choline transporter. Now this is a co-transporter, which means that it is transporting 2 things at the same time. As you can see in the diagram here, choline is transported into the presynaptic neuron along with sodium ion. So 1 choline with 1 sodium ion. Now if you think about this, these co-transporters are useful because it allows another substance to be brought into the cell while using the advantage of the potential energy in concentration gradients. So if you think about sodium and where it's most concentrated outside of a neuron, or it's most concentrated outside of the neuron at rest. And so sodium would be moving down its concentration gradient and also down its electrostatic gradient to move into the cell. Right? And this facilitates the transportation of choline down. Right? So this is a transportation that doesn't require any energy on the part of the choline transporter. But that energy is no extra energy. So this isn't a transporter that requires ATP. It doesn't phosphorylate ATP or ADP. but what it does is it uses the energy that we have in the concentration gradient. Right, so as sodium flows down its concentration gradient, it facilitates the transport of choline back into the cell, and choline is recycled and reused once again to make acetylcholine in that presynaptic neuron. And this is a pretty fast reaction. We can see here at the catalytic activity, the rate. About 5,000 acetylcholines are broken down for every acetylcholine esterase per second. So that's pretty fast. Now let's take a look at a real-world example of a toxin that affects acetylcholine esterase. So in the next few slides, I'm going to talk about sarin gas and the events that happened in the mid-90s in Tokyo, the Tokyo subway sarin gas attack. So this is a story of the infamous Tokyo subway sarin attack that happened in 1995. It was perpetrated by a doomsday cult, Om Shinrikyo cult, which gained popularity in Japan. It was founded in the late 80s by an acupuncturist named Shoku Asahara. It gained popularity, about 10,000 members strong at its height. It still exists today in some capacity, but not as popular as it used to be. They even in the early 90s gained an even greater following in post-Soviet Russia. About 30,000 Russians joined the cult as well. And it was founded on teachings of yoga and mysticism, and had elements of Buddhism, Hinduism, and Christianity mixed in. But in 1995, they perpetrated this attack on the Tokyo subway system during rush hour, where they released sarin nerve gas into the tunnels. They attacked and killed 13. As a result of releasing the gas, 13 people died, and nearly 10,000 were affected in some way. About 50 were very seriously injured. And so what is sarin gas? So it's what's known as a organophosphorus compound that prevents acetylcholinesterase from hydrolyzing acetylcholine. And so you can think, based on what we already know about acetylcholine, right? It's the neurotransmitter at the neuromuscular junction. It's excitatory, so when it's released, it causes the postsynaptic cell, which is our muscle fibers, to contract by opening up sodium ion channels, which in turn stimulate calcium channels to open for muscles to cause its own action potential and contraction. And so without functional acetylcholinesterase, and we saw before, right, this is a very active enzyme. 50,000 acetylcholines are broken down every second by one acetylcholinesterase. So sarin gas renders acetylcholinesterase ineffective, and we have a buildup of acetylcholine in the synaptic cleft, and this causes a constant stimulation of those muscle fibers in the postsynaptic cell, and these can cause very violent muscle spasms and neuromuscular rigid paralysis. And like our other toxins, neurotoxins that affect the periphery and affect the neuromuscular junction, these are always things that cause paralysis, either through rigid paralysis or flaccid paralysis. And the way that this is fatal is by making the individual unable to breathe. They're unable to rhythmically contract and relax their diaphragm in order to respire. And this certainly was the case for those 13 victims of this attack in the Tokyo subway system in the mid-90s. So this is an example of the second way that neurotransmitters are removed by the synaptic cleft, right? Enzymatic degradation. The third and last method is reuptake, the absorption of a neurotransmitter by the presynaptic cell that secreted it. This is the most common mechanism of inactivation of a neurotransmitter. It terminates the signal, it recycles the chemical back into the presynaptic neuron, and it is accomplished through transporters, little molecular proteins in the presynaptic membrane that transport the neurotransmitter back in. Now, this is an example of a specific reuptake transporter for serotonin and a class of drugs that are used to target this molecule. Serotonin, selective serotonin reuptake inhibitors, SSRIs, which we've mentioned in class before. So a neurotransmitter, in this case serotonin, also known as 5-HT, released by the neuron. There are a variety of different receptors that could be in the postsynaptic cell, and the transporters we can see in the presynaptic cell here in green that facilitate the reuptake of serotonin, but they can be blocked by SSRIs, preventing that reuptake. Serotonin remains in the synaptic cleft for a longer period of time, which gives it more opportunity to interact with the receptor. So it enhances that signaling. It enhances the serotonin signaling in the presynaptic or in the synaptic cleft. An example here of glutamate shows us that most neurotransmitters are removed by a variety of different methods in the synaptic cleft. They can include perhaps all three, sometimes diffusion, enzymatic degradation, and reuptake. Here with glutamate, we see two different pathways for its removal from the synaptic cleft. We see reuptake back into the cell, and we also see enzymatic degradation. We have the EAAT, which stands for the excitatory amino acid transporter, which can transport glutamate directly back into the presynaptic cell, or often it's taken up by a neighboring glial cell. Inside the glial cell, it'll undergo a chemical modification. Glutamine synthetase can chemically change it to glutamate to glutamine, and glutamine will be transported out of the glial cell by the SN1 transporter, and this stands for sodium neutral amino acid transporter, and it can be taken back up into the presynaptic cell then by the SAT2 transporter, and inside the cell, glutamine can be chemically modified and changed back into glutamate via the glutaminase enzyme. Now, we have neurotransmitters in two very broad categories. We know that there are over 100 different types of neurotransmitters that we have been able to characterize so far. There's likely, it's been estimated twice as many, so over 200 that probably exist. And they can be separated into two broad categories. First, and highlighted on the bottom, we have neuropeptides, or peptide neurotransmitters. And they are composed of relatively large proteins, usually 3 to 30 amino acids. And this category, as we find, has the lion's share of the different types of neurotransmitters. And the other one are small molecule transmitters. And these are typically individual amino acids or biogenic amines. We've talked about some of these already, such as acetylcholine, glutamate we just talked about. Also, very important neurotransmitter GABA, which we'll talk about. ATP can be used as a neurotransmitter. And certainly ones that you've heard of before, such as dopamine, serotonin, histamine, epinephrine and norepinephrine, which also go by adrenaline and noradrenaline. And we'll talk about some of these individually shortly. Acetylcholine is a neurotransmitter we've talked a lot about in the context of the neuromuscular junction, where it's a excitatory neurotransmitter that causes a depolarizing postsynaptic potential, right? Or an excitatory postsynaptic potential, EPSP. But it's found in other places in the nervous system as well, and we'll go over those briefly too. And we'll start using terms that have to do with, that characterize the association with or actions of or a similarity to specific neurotransmitters. And this is the first one for acetylcholine, cholinergic. So neurons that release acetylcholine, or we can call cholinergic neurons. Receptors that bind acetylcholine we can call cholinergic receptors. A synthetic drug that mimics the pharmacological actions of acetylcholine we can call a cholinergic. So anything that's associated with the production, release, the binding of acetylcholine we call cholinergic. Now, acetylcholine is also found in the peripheral nervous system and the autonomic nervous system, not just the neuromuscular junction. It's a neurotransmitter for all of the preganglionic nerve fibers of the autonomic nervous system, both sympathetic and parasympathetic. Now, we likely mentioned this very briefly when we first talked about the autonomic nervous system, but didn't get into too many details. But as a reminder about what pre- and post-ganglionic means, if you remember, our autonomic nervous system is typically made up of fibers that are coming from the central nervous system into the periphery, and they synapse somewhere in a ganglia. A ganglion is a collection, if you remember, a collection of neuron cell bodies in one area, and they can be found in different locations in the periphery, and then preganglionic neurons synapse there at the ganglia, and then the postsynaptic ganglion or the postganglionic neurons are the ones that extend to the effectors or the cells, tissues that are the target of these fibers. All right, in this image here, we're taking a look at the sympathetic nervous system, and you can see in red are the preganglionic nerve fibers that extend out either into the sympathetic chain ganglion, which is right next to the spinal cord, right outside the vertebral column, or there are a handful that extend further out into the periphery, and they will synapse in ganglia that are called the prevertebral ganglia. And so blue here would be the postganglionic nerve fibers, and red are the preganglionic. So those red nerve fibers are the ones that are producing and releasing acetylcholine. And in that last image, the blue ones are typically releasing norepinephrine. And this is an image of the parasympathetic nervous system, and it's the same here as well. These green fibers are the preganglionic fibers. And the orange ones, which may be hard for you to see, but that's because they're right very close to the effector tissues. Those orange ones are the postganglionic ones. So the ganglia for the parasympathetic is typically much, much closer to the effector cells. But these green ones, like in the sympathetic nervous system, also release acetylcholine. And there are a few areas in the brain that also release acetylcholine, some very specific nuclei in the brain. The basal forebrain, part of the forebrain, releases acetylcholine, and their fibers extend throughout the cortex, also to the amygdala and the hippocampus. And there are nuclei in the brainstem that produce acetylcholine as well, extending to other areas of the brainstem, including the cerebellum. Their function in the brain is being involved with arousal and attention, and also are important part of the network that's responsible for learning and memory. It has an important role in the enhancement of sensory perceptions when we're awake and engaged in sustained attention. Now let's talk about some acetylcholine receptors, of which there are different types. We have nicotinic acetylcholine receptors. All right, so in the shorthand is NACHR. These are ionotropic receptors, and so you should know exactly what that means. Ionotropic, these are ones that are of themselves channels. So the receptors that bind acetylcholine are also a channel. And these are cation or positive ions channels. And it has two binding sites, and it generates an excitatory postsynaptic response, EPSPs. And it also binds, as you might imagine, because of its name, nicotine. And nicotine use results in euphoria, relaxation, and also is highly addictive. And this here is a good time to mention that neuroscience has a rich history of finding new things in the brain through exogenous chemicals. And so you can imagine perhaps how did we find this nicotinic acetylcholine receptor? It wasn't through experiments with acetylcholine. It was through experiments with nicotine. We've known for a long time that nicotine has a profound effect on perception and cognitive abilities and activity. And so we know that it must be doing something in the nervous system if it's affecting all these things. And we eventually found out what characterized the receptor that nicotine binds to in the brain. And we called it the nicotinic receptor. Later, to discover that the nicotinic receptor also binds acetylcholine. And so every exogenous chemical that we put in our body that has an effect, it's binding to a receptor in the nervous system. So therefore, there must be an endogenous chemical that is the same or very similar to that exogenous chemical. Something that our body produces must be there, because why else would this receptor evolved and be inside of our body? And so, as we'll see, many of the receptor names that we have in our nervous system are named after the original exogenous compounds that were used in their first discovery. The second type of major acetylcholine receptor are what are known as muscarinic receptors. These are metabotropic receptors. Unlike the ionotropic nicotinic receptors, they aren't in of themselves a protein or an ion channel, but rather they're G protein coupled receptors. So they are the start of a cascade of intracellular signaling in the postsynaptic cell. And there are 5 different subtypes of muscarinic receptors, and they all result in slow postsynaptic responses. Some of them are inhibitory, some of them are excitatory, just depending on where they are and what type there are. And they're very simply labeled M1, M2, M3, M4, and M5. And the muscarinic receptors, they mediate most of the effects in the brain, most of the cholinergic effects in the brain. And they have, among other things, inhibitory effects on the dopamine-mediated motor pathway, and they have excitatory effects in the hippocampus. Now, why are they called muscarinic receptors? And that's because they were experimenting with muscarine, a toxin, when they discovered the muscarinic receptors. So the acetylcholine receptors, the specific type, M1 through M5, they bind to a toxin called muscarine that's found in a handful of different mushroom species in the genera Inocybe and Clethocybe. And they affect muscarinic receptors in the periphery because muscarine does not cross the blood-brain barrier generally. And so it has a lot of effects in the periphery. There are muscarinic receptors in the periphery, in the autonomic ganglia, and these muscarinic receptors mediate the inhibition of heart rate via the parasympathetic nervous system. So they're part of the parasympathetic activation. And this ingestion of the muscarine toxin results in a number of different effects, such as meiosis, not the meiosis that's involved with cell division, but with the meiosis that means excessive constriction of the pupils, sweating, salivation, lacrimation, which is excess production of tears, bronchoconstriction, so this is the constriction of air pathways, bradycardia. This is the abnormally slow rate of your heartbeat, abdominal cramping, and it can even lead to cardiac arrest, so it can actually stop your heart. If it gets into the brain, it can cause convulsions and hypothermia, which is sort of a strange symptom. The toxin from these poisonous mushrooms can actually lower your body temperature. And so I want you to maybe hold on to this, and we'll think about this again when we start talking about the central nervous system anatomy, different parts of the brain. Keep this question in the back of your mind. Where in the brain are muscarinic receptors that would influence hypothermia? All right, so once we start learning about different parts of the brain, I bet you'll be able to predict where you would find muscarinic receptors because of this symptom it has of hypothermia. Now let's talk about some pharmacological uses of muscarinic receptors. Atropine. This is a drug that is a competitive antagonist. So this means that it binds to the same site as the agonist, which is acetylcholine, but does not activate the receptor. So it's competitive. It binds to the same thing as acetylcholine, which is the muscarinic receptor. So it competes with the neurotransmitter. It's an antagonist. So that means that when it does bind to the receptor, it does not activate it. So as long as atropine is bound to it, acetylcholine or nothing else, no other cholinergic compound could bind to the receptor. And so basically that receptor can't be used. It's inactivated as long as atropine is bound to it. Agonist would be anything that would activate the receptor. Antagonist is anything that does not activate it when it is bound. Atropine. This is a chemical that's found naturally in a handful of different plant species, including a deadly nightshade, which is a common name that a lot of people have heard before. And it's from the plant Atropa belladonna. Atropa is the genus name. You can see here that atropine was named after the genus for the plant that it was extracted from. This is used in surgeries to reduce saliva. So it has actions in the periphery, in the autonomic nervous system, to reduce saliva. And so a lot of anesthetic cocktails will have a little bit of atropine in there to reduce saliva. It can also treat bradycardia, so that's slowing of the heart rate. And it can be used as a treatment for nerve gas poisoning, such as sarin gas. And if you remember when we talked about the effects of sarin gas, it basically overactivates the postsynaptic activation due via acetylcholine, and it does so in sort of the roundabout way, right? It prevents acetylcholine esterase from breaking down acetylcholine. So acetylcholine stays longer in the synaptic cleft, and it builds up with repeated signals. And so that postsynaptic target cell, whether it be muscles, skeletal muscles, or other effector target cells, tissues in the periphery, they get overactivated. And so the atropine is a competitive antagonist. So this buildup of acetylcholine, a little bit of atropine, can help compete with acetylcholine, rendering essentially many of the receptors unresponsive to acetylcholine because they've filled up the spot and acetylcholine can't bind to the receptors anymore. What would an ophthalmologist use it for? If you remember in the last slide, one of the effects of muscarine was miosis, which is the constriction of the pupil. And so actually what it's used for is to counteract that miotic effects of the muscarinic receptors. It activates pupil dilation, or the medical term mydriasis. And so if you've ever seen the eye doctor and they've given you a drop or two in each eye to make your pupils really big so that they can get a nice good look at your retina behind the pupil, this is what they've used, atropine. Now we're going to talk about psychotropic effects of muscarinic receptors. Psychotropic means, or is a term that means relating to drugs that affect a person's mental state. The first one we're going to talk about is scopolamine. Scopolamine is a muscarinic antagonist, and it's used to counter motion sickness and postpartum cramping. And this is a drug that you need a prescription for. It's not over the counter. It is a drug that's naturally derived from a few different flower species from the genera Datura or Brugmansia, and they are commonly known as angel's trumpets, but also known as devil's breath. And perhaps that latter common name is because of the psychotropic effects that it has. It impairs memory, causes amnesia, induces delusions, and is a known hallucinogenic. But not the good kind of hallucinations. Here's an example of a a case study of a group of people that ate some of the angel's trumpet flower, the Datura flower, Datura arborea, 1985. So this was in Australia, Victoria, Australia. And they suffered severe hallucinations, and in fact, one of the members actually drowned in a shallow puddle of water because of the psychotropic effects. And so this is like I said, this is a bad psychotropic drug. It is known known to be used in some indigenous cultures. In ritualistic, shamanistic traditions. And it's used by a very specific class of people or individuals in their tradition that have sort of learned and earned the right or ability to use these for spiritual purposes. And they use it to induce altered states, and they use it to induce a powerful trance, and it can have very violent and unpleasant effects. Even after the psychotropic effects are done, there can be long-lasting side effects. Sickening after effects is described here in the Encyclopedia of Shamanism. And it's been described as causing temporary insanity. And it's thought that the sort of the spiritual practitioners that use this, or the shamans, they're viewed as sort of paying a very heavy price for this sort of spiritual benefits that they get from using these substances to induce altered states. And like I said before, there's usually just a very select few people within these cultures that use it. Then coming up next is another example, another case study of the use of these muscarinic antagonists from these flowers. And this is a little bit of a warning. This one is disturbing, involves self-mutilation. So if you don't want to hear the details, you can sort of skip over the next 30 seconds or so. This case study was published in the European Archives of Psychiatry and Clinical Neuroscience, and the title is Self-Amputation of Penis and Tongue After Use of Angel's Trumpet. And here's an excerpt here from the paper. Approximately 24 hours after the self-amputation, the young man was examined by a psychiatrist from our team. The patient reported a complete amnesia for the time between going to bed, and this was before the delusional and hallucinogenic effects kicked in after he consumed the angel's trumpet. So completely forgot everything between going to bed and waking up from the surgical operation in the hospital to help his self-amputation wounds. No other psychopathological symptoms were registered during the psychiatric investigation 24 hours after the event. So this poor individual has no memory of the traumatic effects of what this substance had on him, just through blocking muscarinic receptors. So how do muscarinic receptor antagonists cause hallucinations? Well, a leading hypothesis for this suggests that that because muscarinic receptor activation in the brain, specifically in the frontal basal nuclei, that project through the rest of the cortex, that they are involved in mediating the narrowing of sensory information and intrinsic internal information that our brain is constantly exposed to. It sort of filters that before it reaches our conscious level. So we're constantly getting in, for example, stimuli from our environment through all of our five senses that we never are conscious about. They don't reach a level of salience, which just means the sort of important stuff, which is good, because otherwise it would be very distracting and very hard to concentrate on things. And you may have experienced this before. If there's sort of like a noise that's fairly consistent, our brain does a good job of filtering it out. And you may have sort of from time to time sort of kind of become aware of a noise that was like a fan or traffic or a TV in another room, that you sort of like realize, oh, that's been playing the whole time and I haven't been consciously aware of it. And so this is through the actions of, in part through muscarinic receptor activation in the cortex. And so most of the information that comes into our central nervous system remains at the subconscious level. And that information gets filtered out before it gets into our conscious stream. Inactivation of muscarinic receptors in the brain, it's hypothesized that it causes a breakdown of that filter. And that parallel stream of subconscious information and processing is allowed to sort of like flood into the conscious awareness. And our conscious awareness has a difficult time dealing with all of that sort of sensory overload. And as I've mentioned before, our brain likes to, it takes in all the information from the environment, and it creates a story, a narrative that consciously makes sense to us because of how we develop and how we learn. We kind of figure out how the world works. So if we get some kind of sensory information, our brain likes to make predictions about what will happen, what that means, what it's connected to. And so when we get all of this information, our brain is racing to sort of make a narrative, and will actually invent things to sort of try to make something that makes sense, to explain all of this information that's coming in. And it's thought that is why we experience hallucinations. That it's, that phrase, it's all in your head, and it, then that's quite literally the truth. And so this is the leading hypothesis. Muscarinic receptor activation helps to keep the floodgates of unconscious information closed. And when they're wholesale deactivated through pharmacological means of these toxins or these compounds from muscarinic antagonists, the floodgates open, and our conscious is bombarded with all of this unconscious information that we otherwise don't have to deal with. Okay, so we're going to stop there. I'll post another video. We'll continue talking about a few more neurotransmitters, and we'll get things started in the next video talking about glutamate, which is the excitatory neurotransmitter. So I'm excited to tell you about it. I'll see you next time.